Couple of quick announcements. The first one is that I just put a copy of the 6th edition of the textbook on reserve in the library. Probably not immediately available there, but it should be next week. I have a couple of other textbooks that I'll be taking over there as well, including the problems book that goes with the 6th edition. I don't look at that problems book. Some people find it useful, some people do not find it useful at all. But I will be putting it there. I also posted on D2L for the textbook that Garland Scientific, who publishes it, as a special deal, so you can get 25% off of the printed version, and I think the ebook rental is something like $50 for 6 months, so it's not ridiculous. Still expensive, and I couldn't do the calculation to see if 6 months works for this term and next term, but that might be good for both molecular biology and cell biology. So that's all there. The recording of lecture on Wednesday is posted on YouTube. The link is there. I also uploaded an MP3 audio for the lecture on Wednesday. Some people like that. Good, like MP3s. Good, just audio. Yes, a good thing. Okay, so I will try and continue to do that as well. I think that's it for announcements for today. So we'll move right along with our clicker questions. They don't count today, so if you don't have a clicker yet, that's okay. Next week, however, they will start counting. So, almost all polymerization reactions in biology are acid-base catalysis, condensation, hydrolysis, dehydrogenation, or reduction reactions. And feel free to talk about this, but it's fine. It's not an exam.
Very hard. Get back here. Get it. Wait, there again. Yeah.
That's what
Well, hydrolysis is like when you use water. Dehydrated, you take water out.
Twenty seconds. And okay, so we are split about half and half between condensation and hydrolysis reactions. Yes, water has a lot to do with it, but what are we talking about? We're talking about a polymerization reaction. What's happening there?
breaking two covalent bonds.
Okay, so the polymerization reaction is doing what? It's taking monomers and making polymers. So that is the reaction, is taking monomers and making polymers. And so that is the actual reaction. Is that an energetically favorable reaction? Heck no. So what's going on?
Coupling.
Yeah, coupling, yes, true. But that wasn't one of the answers.
Oh, condensation.
So it has to be a condensation reaction. So you're taking water out rather than putting water in. And so that's going to be energetically unfavorable because you're changing entropy, because you're going from something which is less ordered to more ordered, and you're going to be taking water and putting it into a situation where there's already a really high concentration of water. So you're really going against it in two different ways there. So B is the correct answer. Our next clicker question is, you are told that the dissociation constant for protein-DNA interaction is nanomolar. What does this mean? There's a very tight binding between the protein and DNA. There's a very weak binding between protein and DNA. There's very little non-bound protein. There's a lot of non-bound protein. Or, step Stedman's favorite, A and C. Again, feel free to talk. Supposed to be interactive things. Engaging. Okay, there seem to be considerably more differences of opinion on this one. And when I see something like this, I think it probably means I didn't do a good job of explaining it. And interestingly enough, I can see your votes up here at the front, so I see how they all change with time. So the consensus here is A and C, which is correct, by the way. But the important thing here really has to do with what a dissociation constant means that I guess I didn't do a terribly good job of explaining last time. What you're looking at is an equilibrium between two things that are bound to each other and the two things separate relative to each other. And the protein binding is one of those things, the DNA is another one of those things. And the dissociation constant is just looking at how often these things are associated with each other or not associated with each other. At a nanomolar dissociation constant, you have 10 to the minus 9th molar of the non-associated things. So that means you're going to have very small amounts of unbound protein. And that interaction is going to be a very tight interaction because you're not getting that coming on and off all that rapidly. So does that make sense? Not make sense? Etc. Okay. So moving on. Hopefully we covered as much of chemistry as we need to. Again, all in a day and a half of lecture. In fact, one half that you guys did by yourselves. Main important things here again are that it's all about the weak interactions, multiple weak interactions that are reversible. You get enough of them together, you can have something like a nanomolar dissociation constant in terms of interaction between protein and DNA. Water, critical to life as we know it, and it's really the hydrogen bonds that water form that give you the hydrophobic interactions, not strong bonds. Talk a little bit about strong interactions, and mostly, in fact, the strong interactions we're talking about are going to be the covalent bonds that form, mostly due to polymerization, and we'll see this slide actually works. No idea why it didn't. Same computer, same image, but it doesn't make. So the important thing here is really that in order to get polymerization, what kind of a reaction is it? It's a condensation reaction, which means that it's what kind of energy?
High energy.
Yeah, it's going to require lots of energy input. It's going to be energetically unfavorable. So to do that, you need to couple it, as Bill was mentioning here as well, to some other high energy bonds being cleaved. And so you have hydrolysis connected to condensation for the most part. And so here, your loss of water is going to be connected to some other energy. And again, you have to have this coupling which takes place. It's also really important, because I mentioned this a little bit last time, but it's a little bit easier to show here. These have to be irreversible reactions in some way, because if not, all of our DNA would fall apart. All of our proteins would fall apart. All of our RNA would fall apart. So you have to have some way of making that a irreversible reaction, because energetically, you're a heck of a lot better off here with monomers than you are with polymers. So the way this works with nucleic acid polymerization, and we'll talk about this in much more detail when we talk about both replication and transcription later on, is you have ATP hydrolysis. But that ATP hydrolysis is not directly connected to making your polymer. It's this, adding ATP, making your high energy intermediate, and then cutting off not this last phosphate, which is usually what's happening in ATP hydrolysis, but cutting off both of the phosphates. We'll talk more about this again when we talk about nucleic acid polymerization and the nucleic acid structures next week. The alpha and alpha phosphate, beta phosphate, gamma phosphates going from your oxygen residue here. Cut off your beta and your gamma, that gets you pyrophosphate, which is two phosphate molecules hooked together, also by a high energy bond. That gets hydrolyzed again, and it's this two-step process. First release of pyrophosphate, which gives you your polymerization reaction, and then the hydrolysis of the pyrophosphate, which gives you the irreversibility. So it's high energy intermediate, which is what we have here, ATP, which then gets transferred here to polymerizing your chain. Again, this could be RNA, it could be DNA. And then the irreversibility comes from pyrophosphate hydrolysis. So finally got those two figures. Any questions about this at this point? Again, we'll talk much more about the polymerization of nucleic acids as we move along. Okay, so what does the polymerization? What stimulates a lot of these reactions? It's all about proteins, and we'll talk about proteins now and then skip proteins and talk a lot more about them later on in the course. But just some pretty basic aspects about proteins. Again, hopefully most of this is review. Proteins are made up of amino acids. Those are the monomers that come together to make the polymers. They come together through peptide bonds. What you form with peptide bonds, which will give you what's called the primary structure, just a linear connection of amino acids. Also has a molecular biology polarity, so a start and a stop, from an amino terminus to a carboxy terminus. Those then form secondary structures, which are due to hydrogen bonds that form in your peptide backbone. And the two major forms of secondary structure are alpha helices and beta sheets. There are other ones, but we're going to sort of ignore them for most of this course. Those secondary structures come together to form a tertiary structure. And anyone remember from the first lecture what structure means? Function. Yes, so whenever I say structure, you can think function. And that is what gives you your active protein, what's doing what it's going to be doing. Talk a little bit about some of the protein structures. We can talk about protein structures for physical courses. In fact, there's whole courses on structural biology where people talk about protein structures. There are tens of thousands of them. Most of them are made up of these domains. A domain is just a separate part of a protein that forms a structure by itself. And so if it's forming a structure by itself, it's likely to have a function. Exactly. So multiple proteins are going to have lots of domains in them. Many of these have different functions. There's a little bit of confusion. There's also what people call sequence motifs. A sequence motif is just a linear stretch of amino acids that has certain amino acids in it that can give you some ideas about what that sequence might fall into and give you a particular structure and then a function. We'll talk about the leucine zippers, and we'll talk about zinc fingers. Lots of these are motifs. They're sequences, amino acid sequences, that don't necessarily give you a structure which will give you the function, but you can predict what kind of structure or function they're likely to have. Many proteins are present as multimers, so it's just different polypeptide chain. When I talk about a polypeptide chain, it's got one C terminus and one N terminus. So if you have two of those come together, so two N termini and two C termini, that's going to be a multimer that can go from dimers, where you've got two, to I think the largest case that I know of has something like, at least the RNA polymerases have about 20 different individual polypeptides that come together to make one functional protein. We're going to talk a little bit about protein families, and then we'll talk a little bit about modeling and allosteric as we go on. Amino acids, somebody asked me online, A2L, do you need to know all the structures of your amino acids? No, this is not a biochemistry course. You don't need to know all of them. I think in biochemistry you do. But there are a couple of important aspects about the structure of proteins, in particular structure of amino acids, that is going to be really critical here. The first one is that even in any individual amino acid, you're going to have an amino end and a carboxy terminal. So the monomers already have a molecular biology polarity to them. They've got a front end and a back end. They get hooked together through these carboxy termini, makes a peptide bond with an amino terminus. You end up with two that hook together. That's a dipeptide, tripeptide, tetrapeptide. All four of these link together. The chemistry here of the so-called backbone, we've got all the peptide bonds, is absolutely identical. Doesn't matter what that is, with one exception, which is what people take in biochemistry. What's the exception? Proline. Proline actually will look back around. But for the most part, again, 19 out of the 20 normally used amino acids, you're going to have exactly the same kind of backbone here. And then the variable groups that make those 20 amino acids different from each other are those that are coming off here from the middle carbon in any of the amino acids. It's also known as the alpha carbon. There's where you'll have all of your side chains, or also known as R groups. So this is what it looks like in the chemical state in the middle. Usually people will draw them as cartoons, and basically just the sequence. Again, I don't expect you to know the names of all the amino acids. I don't expect you to know the structures of the amino acids. If I give you one, I will let you know. Occasionally, however, I will slip into using the single letter abbreviation for a lot of amino acids, just because it's easier and it's also what everybody uses in the literature. So, but again, you don't need to know them all, write them all down for you. There are, of these, normally 20 used, there are actually about 23 amino acids that are used in proteins, but the extra 3 are really interesting, cool cases that we don't have a chance to talk about in this class. But they're really sort of 3, and some people will call them 4. I like to call them flavors of amino acids. But it's basically the differences that you have in each of those R groups, each of the side chains that are hanging off of it. You've got the nonpolar amino acids, and these are guys that basically are not forming hydrogen bonds. All over here on this side, the most important of these is going to be the leucine. We'll see that for leucine zippers and coiled coils in just a second. There are a number of polar amino acids, particularly important for cell biology signaling, which we're not going to talk that much about, are those that have OHs on them. This is the serines, threonines, and tyrosines. But as far as we're concerned, the most important structurally, and also really important in terms of interactions with proteins and nucleic acids, because, of course, nucleic acids is where it's at, the proteins are boring, have to do with these charged amino acids, the aspartates, glutamates, arginines, and lysines. And so why is this so important? That's because at physiological pHs, which is around pH 7, very often your acidic amino acid side chains have given up a hydronium ion to water and will be stably negatively charged. And on the flip side, the basic amino acids, like lysine, have a normal positive charge because it picked up a hydrogen ion from the water that they're surrounded by. And what that means, of course, is that these guys, like these guys, because these are they're oppositely charged relative to each other. So this is very important in terms of protein structure. These charged side chains will be interacting with each other, but also those charged side chains will be interacting with other things that are charged, like water and What else will we talk about for the whole rest of this course? Nucleic acids. All nucleic acids are incredibly charged. I don't have my DNA model, but don't worry, as of next week, you won't see me without my DNA model. So really, a lot of these charge interactions are very, very important in terms of protein, nucleic acid interactions. So, yes, you don't need to know the structures of all of the individual amino acids, but I would know that arginine and lysine are generally positively charged, and then glutamate and aspartate are generally negatively charged. So, yes?
So I see glutamic acid, aspartic acid, and then glutamate, aspartate named differently. So it's probably a stupid question, but what's the difference between the two and when are they used?
Okay, so glutamic acid and aspartic acid versus glutamate and aspartate, that's going to depend which way they are, so whether they're protonated or not.
Okay.
But basically, I would use them interchangeably as far as this course is concerned. The biochemists may argue with me about that, but as far as this course is concerned, I'm going to use them interchangeably with each other. Good question. Oh, yeah.
As far as when proteins fold, do you mostly just have to worry about rotation in the bonds in the backbone, or are all the side chain bonds part of how it can fold?
The question has to do with protein folding and what do we need to worry about in terms of thinking about protein folding. How do you get proteins to come together? We'll get to that in just a second, but the answer is basically yes to the first and no to the second. So, yeah, so the question is, how do you go from this linear sequence of amino acids that are all hooked together, you've got peptide backbone with all the side chains sticking out from it. How do you go from this linear sequence of individual amino acids to something that actually has some kind of function? So to do that, you've got to get structure. So we go from our primary, also people talk about the primary structure, which is just your sequence. By convention, everyone goes from amino terminus to C terminus. Those sequences of linear sequence of amino acids almost always are going to form into some kind of secondary structure. And these secondary structures are all due to hydrogen bonds that form between backbone atoms. So any kind of second structure is backbone atoms. How those backbone atoms then fit together to give you a three-dimensional structure, that's where the side chains come in. And so it's where all the side chains are interacting with each other, or side chains interacting with water, which will then give you your final structure. Now, this may seem a little crazy to be talking about, okay, why do we talk about primary, secondary, tertiary, and when you've got multiples that come together, quaternary structures, that's the way these things are made. The amino acids are polymerized by the amino acid polymerase, what other people know as a... Is that early in the morning? Ribosome. So the amino acid polymerase is a ribosome. It makes these linear stretches of amino acids, which of course are totally worthless unless they actually have a structure. And so this process of going from linear stretches of amino acids and getting into that final structure that gives you a function is absolutely critical. And people have been trying to figure this out for basically as long as we've known about proteins. And we still don't have a good handle on how it all fits together. Getting better, but still it's very, very hard to predict how all these things are going to come together. But when people look at these final structures, then you can go back and say, oh, we've got all these interactions which are causing, or appear to be causing, that structure. So here we've got, in the middle of your protein, away from the water, not surprisingly, these are things that are not forming hydrogen bonds with anything. So water forms lots of hydrogen bonds, forces them together. So very often the middle of proteins, you're going to have a lot of the hydrophobic side chains or the nonpolar side chains. And on the outside, you'll have a lot more of the polar ones. Yeah?
Is that a representation of the quaternary structure or the tertiary structure?
Down the bottom?
Yeah.
Yeah, so this is going to be a tertiary structure. You only have a quaternary structure when you have multiple polypeptides associated with each other.
Okay. So that's happening because of interactions between the secondary structure and its aqueous environment?
So the reason you get a tertiary structure is how you go from secondary structure to tertiary structure? Mm-hmm. Yeah, so how you're getting this secondary to tertiary structure is interaction of the side chains with each other and with water, and in many cases also other proteins.
Sure.
You consider three-dimensionality happening at tertiary or secondary structures?
So there's a little bit, so the question is, sorry to repeat here, but do I consider that you're getting three-dimensional structures at secondary or tertiary? You are in fact getting some three-dimensional structure at secondary, and we'll look at those, but generally that is not going to be something which is stable, and it's when those secondary structures come together is usually what you think about as tertiary structure. But if you're going to be nitpicky about it, then these secondary structures do have some kind of three-dimensional structure, but even the linear structure does too. So, but in terms of function, it's going to be those secondary structures coming together. Sometimes it can be really simple. We'll look at some of those examples in just a second. Sometimes it can be highly complex. Yeah.
Since the secondary structures are not as stable, do you see a lot of dysfunction? Like, is there an issue with folding there that causes a major dysfunction and maybe causes problems down the line?
So I guess the question here, sorry if I'm misinterpreting this, is when you see problems with folding, do you usually see problems in terms of the secondary structure formation, or do you usually see problems in terms of going from secondary structure to tertiary structure? Mm-hmm. And the answer to that is usually from secondary to tertiary, because that's when your side chains are having the interactions. Backbones usually don't change very much. Okay. And we'll talk about this when we talk about protein folding a little later on. They usually find secondary structures pretty quickly. It's that tertiary structure that's the slower step which actually happens. So we just look at this. These are a number of tertiary structures. Some of the authors of the textbook work on SH2 domains, so that's why they beat on them like mad in this textbook. SH2 stands for sarcomology. That's the only time you're going to hear it from me. Don't worry about it too much. But it's a nice domain that has nice secondary structure, alpha helices, three beta strands here. These are just four different ways of looking at tertiary structures. And I just wanted to mention at this point that the movies that are linked to the textbook are actually really nice in some of this, because I love looking at these things in 3D. I can't wrap my brain around these things so much in 2D. But just looking at them in 3 dimensions is really nice way. This is the cartoon form that we're going to use most of the time through this course, which has a cartoon of what the alpha helix looks like, cartoons of what these beta strands look like, and then these little loops on the outside. This is what the protein would really look like if you're actually interacting with it, because these are the van der Waals radii, so this is the optimal position that you would have in terms of getting your attractive forces. You remember the bump that we saw last time in our van der Waals interactions. Let's look a little bit at some of these secondary structures. Probably Portland's really scientifically most famous son, Linus Pauling, was the one who came up with this, basically through model building and looking at various different proteins, both discovered the alpha helix and the beta strand. But all this is looking at hydrogen bond interactions, and Linus Pauling is also credited with coming up with the concept of the hydrogen bond, where you have interactions between the hydrogen, which is on the amino group, and the oxygen, which is on the carboxy group, of amino acids in your peptide backbone, separated by 4 amino acids relative to each other. And as you remember, hopefully from last time, these hydrogen bonds have to be quite close to linear interactions between each other. So just wrapping around 4 amino acids, you end up lining up your amino group and your carboxy group of each of those. And in that way, you can make a very stable structure. But the really neat thing, as far as I'm concerned about this, is that, yeah, it's a nice stable structure, but what happens to all of your side chains? All the side chains in an alpha helix are sticking out of this alpha helix. So if you've got an alpha helix, it means all of the information, all of the differences that you have between each of those individual amino acids is sticking out. The alpha helix is holding things together, it's a nice stable structure, but then all the information, all of those amino acids are sticking out around the outside. Beta sheets also have hydrogen bond interactions between amino groups and carboxy groups, but now it's not 4 amino acids away in your linear sequence. It's another sequence which is separated by quite a long way. And it can be actually a really, really long way in terms of your primary sequence. These are now hydrogen bonds that form between different primary sequences. So here, so one strand, hydrogen bonds form with another strand and another strand. And as long as you have a lot of these lined up next to each other, you end up with what's called a beta sheet. Originally found also by Linus Pauling, also in studying various proteins, in this case it's the silk protein that he found that had these particular structures. Yeah.
I was just going to say, is there like a chemistry reason why they're using alpha and beta, or is it just because?
Oh, why alpha and beta?
Yeah.
Because it was Linus Pauling, he found the first one, he found the second one.
Yeah.
That's the only reason, as far as I know, for alpha and beta. So that has nothing to do with alpha carbons or beta carbons or alpha phosphates or beta phosphates. No, it's just first one, second one. And after all, if you're classically trained, that's what you use in Greek. So, anyone had Greek, by the way, in class? Good. I meant to be careful. I made a mistake in Greek. First time I taught this class and got beaten up for it. So,
Don't worry about it.
pardon?
Nobody speaks it anymore.
Yeah.
Same with Latin. Nobody cares.
Yeah. I had Latin in high school. Yes, I'm old. And that's it. But yes, alpha and beta just had to do that. But because of this, you can form also really quite stable interactions, but it's a long-distance stable interaction. But also, just like you have with the alpha helices that are sticking out the amino acid side chains on either side, now you've got amino acid side chains sticking out up and down. And so also, you have the availability of those amino acid side chains to interact with other amino acid side chains or other proteins, with water, etc. And so here, it's a little hard to see here, that's why 3 dimensions is better. Here's one of your R groups sticking up, here's one sticking up, here's one sticking up, they're all sticking up. These 3 here are all sticking down, they're sticking out on the backside. You can have this, what's called anti-parallel beta sheets, again going from N to C termini, as you have here. That's what's in fact shown in this image. But you can also have parallel beta sheets really easily. The hydrogen bond interactions really make no difference on the orientation from N to C or C to N. We'll see some examples of both of those.
How would you chemically try to make one side preferred over the other?
How would I chemically go about trying to make one side preferred over the other? People try and do what's called protein design to figure that out. Basically, it has to do with the length of the loops you have in between these 2 pieces. So if you have a really short loop, the only way that you can form these beta sheets is going to be having it be anti-parallel. If you have a long loop, then they have the opportunity to form these other sheets, parallel ones. But it's a very difficult thing to do. Protein design is hard, and people have been trying to do protein design for long periods of time, not really very successful, particularly with beta sheets, interestingly enough.
So we're not going to be asked anything on it. Probably.
Hey, great question. How would you design one of these? That's going to be really hard to do with the multiple choice question. So you have the secondary structures. How do they form tertiary structures? Mostly through, again, all of these weak interactions that I beat on last time in lecture. Van der Waals interactions, really close packing interactions, hydrogen bonding interactions, ionic interactions here. Again, I don't like the term bond. Very often people will also talk about salt bridges here. And a salt bridge is just the interaction between a stably negatively charged and a stably positively charged, like this acidic and basic residue that you have right here. And of course, hydrogen bonds love to form with water. So if you have some amino acid side chain that's going to interact with water, it's going to be on the outside of your molecule, and that's what these little red stripes are supposed to represent here on the outside. We, again, basically since the discovery of protein structures, have been trying to figure out when you go from linear sequence to something like this. In a few cases you can predict it, in most cases you can't. I'm going to do a quick diversion here into something near and dear to me, and we'll see, of course, much more so with nucleic acids, which is Ulysses. But before that, I'll try and answer your question.
Sorry. So are the salt bridges not shown on this previous image?
Which one here?
Yeah, you said something about a salt
So
bridge.
so this ionic bond here, that's a salt bridge. So it's just a different term for the same thing. So helices. Helices are absolutely wonderful, not just because they form virus structures, because that's of course the most important thing ever, but also because a helix is something which is really easy to form with relatively simple interactions. And it's also going to be really important in terms of nucleic acid structure. Also, why Linus Pauling came up with the idea of the alpha helix in the first place. If you have one molecule that's got pretty similar binding sites on it, this is going to want to interact with another monomer with similar binding sites on it, which is going to want to interact with another monomer with similar binding sites on it. And you can either, if you arrange things absolutely perfectly, get these ring structures that happen. But usually, just by lining up these guys, so this monomer here has identical interactions with this monomer, which has identical interactions with this monomer, which has identical interactions with this one and this one and this one, etc. Going all the way along. And so you can have one structure, relatively small for your monomer, which will give you a larger macromolecular structure just by having these repeating interactions relative to each other. This can happen either in a left-handed form or a right-handed form. I spent a lot of time talking about left and right-handed helices. One year I actually gave people extra credit for bringing me left-handed helices. So I have a collection of, anybody ever use one of these things? What's this for? Old enough to know this used to connect your phone to things. But yes, so I've got a couple of examples of these. You can come down and take a look at them after class. But there are a couple of different ways of looking at these helices. I like to do right arm and left arm helices. So we'll start here with the right-handed helix. I just take my arm and wrap around it. That's going to be my right-handed helix. Take this arm and wrap around it. That's going to be my left-handed helix. So, and you can, again, we can play around with these and take a look at them. Turns out that we've got a lot of right-handed helices in proteins, got a lot of left-handed helices. Talk about nucleic acids. Okay. Why do we care so much about these? Of course, they're great secondary structures, but there are a couple of things that I want to talk about in terms of how very specifically some of these things interact with each other, and really quite commonly. Probably one of the most common of those is alpha helices, which interact with each other through what are called coiled coils. So the coil itself is an alpha helix, and that it's coiling around another alpha helix is that secondary coiled coil. The reason that this works so well is that, as we said, when you have your amino acids, side chains are sticking out on the outside, and every 4 amino acids, you're having this hydrogen bond, which is holding the helix together. That means, actually, when you look at the side chains, every 3.5 amino acids, if you've got a hydrophobic side chain sticking out, every 3.5 amino acids, all those hydrophobic side chains will be sticking out on one side of the helix. We've got another helix that does the same thing. These two guys can come together. And this happens very commonly in biology, particularly for long fibrous proteins. You have these coil-coil interactions between 2 alpha helices, in some cases 3 alpha helices, that just wrap around each other and are interacting because there's lots of hydrogen bonds forming here on the outside, which are pushing the 2 hydrophobic residues into each other. This is also one example of these coiled coils called the leucine zipper, because you have leucines, large hydrophobic side chains, every 3.5 or usually 7 amino acids relative to each other. These come together. A leucine zipper, because if you see every 7 amino acids, you've got a large hydrophobic residue. That's a sequence motif that you can see, because you're just looking at the linear sequence. You can predict now that that's likely to be involved in some kind of coil-coil interaction, just by looking at the sequences. So looking at sequences, we can do sequencing. We'll talk more about sequencing later on. You can see where you have patterns of amino acids. Those patterns of amino acids, if you happen to be every 7 amino acids, is a large hydrophobic side chain, highly likely to be involved in one of these coil-to-coil interactions. coil-coil interaction, those two together may give you a stable functional structure. In many cases, that stable structure is a dimerization structure. They're two different polypeptide chains that are coming together. And so this leucine zipper interaction or the coil-coil interaction is bringing the two of them together. But not necessarily. Very often these coil-coils be part of other pieces of proteins. I mentioned domains before. All that a domain is a separately folded part of your protein. So an example over here, just the different colors are different domains. So here, this sort of gray structure is a domain. This purple structure here is a domain. This red structure is a domain here. Since each of those domains have a particular structure, they're likely to have a particular function. Exactly. And so that's very often the case for all of these proteins. Some proteins will have a single domain, and people actually talk about the single domain proteins. Many of them have multiple domains in them, and it's that interaction of these domains with each other which gives you a lot of diversity. And so if you look at the human genome, it turns out that there aren't very many different domains in our sequences relative to other organisms that have these sequences in them, but the mixing and matching of these domains is quite different, and there's more mixing and matching in the larger organisms, as my friends always like to say. Forget more complex or higher organisms, just larger organisms. So that mixing and matching is really quite common. What do some of these domains look like? They can basically be anything you can think of, and people have found lots of them. All alpha helix, all beta strands, or some mixture of both. And I like this example as well, just from the textbook, because it gives you nicely. Here's an example of where you've got anti-parallel strands. Just getting back to the question about how you design one of these. Relatively short loops between each of the strands, whereas here, they're all parallel in the middle, and very often have these alpha helices in between. So it's the distance between one end or the other thereof. So you've got these structures. Structures lead to function. How can you get an idea what the function is from a sequence? It would be great if we could figure that out. If we knew how the sequence went to a structure, and then that structure tells us something about the function. That would be great, but unfortunately, we can't really do that very well. So the next best thing is to now look at two proteins, very often from a protein family that we talked about last time, and look at their sequences, their amino acid sequences. And it turns out, rule of thumb, if the amino acid sequences are more than 30% identical to each other, you'll have an almost identical structure. You've got an identical structure, you have an identical function. So, anytime you look at that, nice example here is elastase and chymotrypsin. They have about 30% identical amino acids. A couple of things that tells you. One is that we actually have a lot of sequence variation and still have a structure and then, of course, function, which is very similar to each other. And it turns out that there are even some cases down here at the bottom. This is a single domain protein here from Saccharomyces cerevisiae and Drosophila. The sequences are less than 30% identical to each other, but it turns out that their structures are almost superimposable, and because of that, not surprisingly, they have basically an identical function relative to each other. So we talked about homology last time, that things have come from a common ancestor. How do we know they come from a common ancestor? If we're lucky, they've got similar sequences, but they might just have similar structures relative to each other. And that similar structure then tells you that these are most likely to be homologous proteins. You can have convergent evolution, but it's a usually much more parsimonious argument to say that they were duplicated and then have diverged relative to each other. Proteins can have all kinds of different sizes, shapes, etc. I like this image just because it gives you relative sizes of things. Here's a relatively small protein, insulin. It's a relatively large protein, glutamine synthetase, immunoglobulin molecule. For comparison, a non-compacted DNA molecule and a non-compacted RNA molecule. Of course, these molecules almost never look like this, but at least it gives you a relative idea of the size of these things. And this is what a lipid bilayer looks like, a standard lipid bilayer. And just by looking comparison-wise, any protein stuck in a lipid bilayer is going to go basically all the way through a lipid bilayer. So this is just an idea, some brief introduction to some of the diversity of proteins and relative sizes. I mentioned quaternary structures before. How do you get a quaternary structure? That's multiple polypeptides coming together. I would say probably the majority of proteins function as multimers of some kind. In many cases, however, those multimers are what we'll call a homomultimer. Homomultimer meaning it's the same polypeptide that's interacting with itself. And so here, we have a dimer of two different proteins. They're two different because they've got amino and C termini, each of them, but they're an identical sequence, and they interact with each other, in this case, by two beta strands, which are hydrogen bonded to each other in the middle of the protein. So dimer, you've got two copies of the same protein, sequence-wise, that are interacting with each other as a dimer. In many cases, you also will have tetramers, four of the same amino acid sequence coming together to form a particular structure. In this case, it turns out that the activity of this protein is dependent on having interactions right here between the two different monomers in terms of activity. Of course, these are really cool examples because they're both from viruses. One of the more extreme cases of a multimer that, again, we'll spend a lot more time talking about later on is the DNA-dependent RNA polymerase. In this case, from Schizosaccharomyces. This is Saccharomyces pombe. The RNA polymerase here, this particular structure has 12 different polypeptides that all come together in order to make this RNA polymerase, which binds the DNA and makes RNA. You have to have every single one of these peptides come together to actually have the final activity that forms from this particular protein. This structure was just solved, I think, about 4 or 5 years ago, and Roger Kornberg got the Nobel Prize for it, basically for solving that structure. So we've already talked about a structure leads to function, but one of the big surprises that's happened in the last few years when people have been looking at proteins, it turns out that quite a few proteins have non-structured regions in them. And it's been really kind of tough to think about this because a non-structured protein, okay, well, how can that have a structure? And we have a structure that's going to give you a function, but how can a non-structured protein actually have function? And why are so many proteins that we look at in a test tube actually not structured? Probably one of the reasons for that is that we're looking at proteins in test tubes. When you look at a protein in a test tube, it's isolated from everything else. And so most of the time, something that doesn't have a structure in a test tube probably doesn't have a structure because it doesn't have the right partner. So in a cell, in a real in vivo environment, a lot of these unstructured, as is shown by this red squiggle, is going to form a structure if it interacts with the right partner, which is just not present in that one particular case. In some cases, however, you really do have stably unstructured proteins present in the cell, and there are techniques for looking at that we'll talk about right towards the end of the course. But those can transiently form structures. And when they form that structure, they have an activity, but sometimes you don't want something to have an activity. So you want something to have activity under some conditions, but not activities under others. And so just changing a structure from something which is having a structure, i.e. having a function, to unstructured and not having a function is a really useful way, and also a really quick way of regulating how things are functioning. Another example here is just extension of that part of a protein, which is structured versus unstructured. Sometimes this will be a domain, sometimes it'll just be a really tiny piece of the protein that won't have a structure unless it's under a very specific condition. And then there are a few cases where actually unstructured is good, and they stay unstructured all the time. And the best example of that is the nuclear pore complex, where you have unstructured proteins which are basically providing sort of, I like to think of this as a sieve, as allowing some things through, but most things not. but it's actually advantageous to have non-stable structures as far as those are concerned. Of course, probably the most famous of the unstable structures are the amyloid proteins. And why do people care about amyloids? Alzheimer's, Parkinson's, etc. And that's because these are proteins that normally are unstructured or they have a different structure that then, under some conditions, can form into structures which are, unfortunately, really stable in some cases, and will accumulate and in some cases lead to really nasty neurodegenerative diseases. So what's happening here is you have these, what are called cross-beta splines. It's just usually one polypeptide that has two beta strands in the middle of it. Those beta strands, we talked about before, they can interact over long distances sequence-wise, or they can interact between different polypeptides. And that process can line up many of these. In fact, so many that you can see them under the electron microscope. And in some cases, actually, the amyloid plaques that you can see in, unfortunately, mostly Alzheimer's brains, they'll see macromolecular structures, which are all made up of these basically little tiny proteins, which have all lined up beta strand next to beta strand next to beta strand next to beta strand. So at first people thought, oh, well, this is just nasty because it's causing cellular problems, etc., in Alzheimer's and Parkinson's. But it turns out that these kinds of structures are also really important for making what are called granules inside of cells. Now, granules, what the heck's a granule? The granule is basically something that microscopists had seen when they were looking at cells. And you look at a cell, now you can see mitochondria, you can see nuclei, but you also see these little blobs. So what's the blob? And so people have studied these blobs in a lot more detail. And it turns out some of these blobs are really important for holding nutrients inside the cell, holding particular regulatory proteins inside the cell. We're talking about splicing components that are being held in some of these granules. It turns out that a lot of these granules are held in one place due to these beta splines, which are bound on the outside of them. And it turns out that in this case, unlike the Alzheimer's and Parkinson's, these are really quite reversible. And so you can get these amyloid formations, these are the good amyloid formations that can happen here on the outsides of a lot of these granules, and just binding to proteins that very often will have lots of repeats on them, or many copies of one particular protein, many copies of one particular RNA, and just holding them in one place in a cell until you actually need them for some particular purpose, and we'll talk about some of those purposes a little bit later on. Very often, these proteins that are going to form these beta splines have what are called low complexity domains. What's a low complexity domain? It's just stretches of amino acids that are repeated multiple times. And not surprisingly, they're repeated multiple times. You see those repeated sequences are the ones that are now going to form up your two beta strands. And often, again, just looking through protein sequences, you'll see low complexity regions, repeated amino acid sequences. Many times they'll form these kinds of structures. Not always, but in many times they will. Now let's talk about some more specific structures. Enzymes, talked about enzymes last time, they lower activation, energy barriers between reactions and products in chemical reactions. They accelerate chemical reactions. Basically, we wouldn't be alive if we didn't have enzymes because none of the reactions that we do would actually proceed with any particular speed. The reason that enzymes work so well is they bring together amino acid side chains for the most part, which then will give you a very specific combination of chemical interactions. There'll be your hydrophobic interactions, there are going to be van der Waals interactions, etc. Give you a very specific place in your protein where you get binding. You get very specific binding, and this binding can be so specific that change of one hydrogen atom is going to give you different binding to one particular protein. So here's an example of an enzyme with multiple amino acid side chains that are now very separate in terms of the primary sequence, but in the tertiary sequence are right next to each other. And that tertiary sequence being right next to each other is then really important for catalyzing some kind of reaction. And in this case, it's a cyclic AMP reaction. It's not critical exactly what the reactions are. But the idea here is that in most of these protein enzymes, most of your amino acid sequences have nothing to do with the activity of this particular protein whatsoever. They're all involved in bringing the few amino acid residues together that will be critical for binding and then the activity of this particular enzyme. So that's basically it for enzymes. Want to know more about enzymes? Take biochemistry. Just wanted to cover one more thing in terms of protein structures, and this has more to do with changes of protein structures. We're going to spend the last 10 minutes of the lecture talking about and how do you change structures. And how do you change protein structures? Well, you change structure. What are you going to do? You're going to change function. So, very important, having particular functions. You're going to be able to turn some of these things on and turn some of them off. There are multiple different ways of doing that, but one of the best studied is something called allosteric regulation. And all that allosteric means is that you have interaction of one thing at one place causing a change in the structure somewhere else. And so in this particular case, this is the example of hexokinase. It's not really that important. But we have one molecule that binds to your enzyme. If it binds that enzyme, it changes the structure, and that structure now allows you to bind to glucose. Now this is active. Again, in this case, hexokinase. What it does is it takes glucose and adds a phosphate group to it. Really critical in terms of glycolysis. In the absence of this other molecule, this structure stays like this most of the time. It's staying in this structure most of the time. It's not going to bind to glucose. It's that binding which is really critical for the enzymatic activity. You're going to have very low activity. But it's not binding, this other molecule is not binding the same place as the glucose. You could also think about particularly cases like this one, where you have a protein which inactivates, a ligand which inactivates one particular protein. This could bind in exactly the same place as your reactants would in an enzyme. This would be blocking at the same place. This would not be an allosteric interaction. However, you can interact somewhere that's away from the binding site for your second ligand, and that gives you an allosteric interaction. So we'll talk more about this a little bit later on, but it's also really important for enzyme regulation. More important for this course is what happens in terms of changing structure for signaling molecules. And those signaling molecules are really basically two, called them flavors, two kind of basic kinds. There's really phosphorylation and GTPases. Now, phosphorylation is a really wonderful way of changing structures because you take a phosphate, so it's not just phosphorus, it's a phosphate, and phosphate means it's got lots of charged oxygen associated with that phosphorus. Putting a lot of charged residues onto something that's otherwise non-charged is going to change the interactions it has with water. It's going to change interactions it has with other parts of the protein. So putting on and taking off of phosphates is a very common way of regulating structure, which of course is then also going to regulate function. Another way that cells evolved to change structures is using what's called a GTPase or a GTP binding protein. These are also known as G proteins. Probably, I think the last time I saw, something like 80% of prescription drugs actually interact with G proteins. And the first ever oncogenes causing cancer is actually G proteins as well. So these signaling proteins are rather important. What happens with one of these signaling proteins, instead of adding a phosphate and taking off a phosphate, which is in fact a covalent modification that takes place. Here, you have binding of GDP, which has one particular conformation, or you're bound to GTP, which is a different conformation. Now we'll look at these in a little bit more detail in the next 5 minutes or so. So, what does the job of putting on phosphates? What's called a kinase. I've got an ace at the end. It means it's some kind of enzyme. Usually, the ace at the end means it's doing something to whatever's in front of it. So, phosphatases, they deal with phosphate. If they're cutting them off, most aces are going to be stimulating some kind of hydrolysis because they're all enzymes. Kinases just put phosphates on. So, in this particular case, we have a serine with an OH. Phosphorylation only happens on OHs. That's why I mentioned the tyrosines, serines, and threonines. We talked about the amino acid side chains a little bit earlier on. Protein kinase will put on a phosphate. The phosphatase will take it off. And usually, when you're phosphorylated, it's an activation when you have a phosphatase that deactivates it. But there are also examples that go around the other way. Some of the largest protein families that we have in the human genome are these kinases, about 500 different kinases that we have in the human genome. GTPases, it's still all about that charge on phosphate, but now instead of covalently attaching it or removing it, now the attachment is to GTP, GDP, a guanosine residue, and that change of having that phosphate or not having that phosphate changes the structure. So here's that example I was telling you about. Oncogene, this is in fact the RAS oncogene, bound to GTP. This helix is in one particular place. As soon as you lose this phosphate, that helix moves. We have a difference in the structure. That difference in the structure then leads to different interactions. And in the particular case of RAS, where it's been modified in oncogenes, that structural change doesn't happen anymore. So that one particular mutation which leads to that. It's a little bit different in terms of how these GTPases work. Instead of putting on a phosphate, taking off a phosphate, that's pretty straightforward. But here, you have GTP, which is bound. Then hydrolysis takes place. And that hydrolysis happens basically spontaneously. You remember all of these triphosphate bonds here, they're relatively high energy bonds. This GTP hydrolysis is going to take place spontaneously. The speed of that hydrolysis can be modified, and we'll talk about some of the modification proteins a little bit later on. But basically, instead of having a switch like you do with a kinase phosphatase, the switch goes on when you put on the phosphate, it comes off when you take off the phosphate. It's much more like a timer. So you have GTP bound, and after a certain period of time, basically, this protein is going to turn itself off. It's going to have hydrolysis that takes place, it's going to be changing the structure, and then to reactivate, it's not a covalent change, it's a non-covalent interaction. GDP has to be removed, and GTP has to be put back in. So just to finish up, last example of this. This is a case of one of the translational regulation proteins. Here we have GTP bound to, in this case, it's a tRNA. As soon as you have hydrolysis, you have a change in the interaction of these domains. It's easiest to see here with the blue domain right here. As soon as the blue domain dissociates from the green domain, you lose this particular activity. Instead of talking about ubiquitin ligation, we'll talk about that on Friday when we talk about, finally, DNA structure. Some of you may have noticed that the syllabus is now getting a little bit off. I will be going back and recalibrating that this afternoon, so we'll have a bit better idea when we're going to be talking about certain topics. Have a great weekend. We'll see you on Monday.